Hi everyone. In this video, we are going to be talking about genetic variation. And I think the full importance of this genetic variation won't become totally apparent until we start talking about things like inheritance and natural selection and evolution later in the semester. But since this genetic variation can be traced back to processes that occur during meiosis and sexual reproduction, I thought it made sense to introduce them now while meiosis is still fresh in our minds. So at this point, we can think about it this way. So if you have genetic or biological siblings, you'll know that there are differences between you and your sibling, right? And at least some of those differences come from genetics that are different between you, unless you're an identical twin. So there are genetic differences between you and your sibling, even though the source of your genetic material, your parents, is the same for both you and your sibling. So how does this come about? How do we get the same parents producing offspring that are different from one another? That's the question that we'll be answering, or at least partially answering today, by talking about three different mechanisms that occur during meiosis. And those three different mechanisms are crossing over, independent assortment, and outcrossing. Mostly the first two are a more direct answer to the question. Sorry about that. So before we get into this, I did want to recap the process of meiosis really quickly, just because the first two mechanisms, crossing over and independent assortment, both have to do with homologous chromosomes. So I want to make sure that we're clear about the role of homologous chromosomes and what happens to them during meiosis before we dive into this. Hopefully, you recall that the start of meiosis is a diploid cell, a diploid parent cell. And in this diagram, we can see that we have two pairs of homologous chromosomes. So we've got a larger chromosome, and we've got two versions, a blue and a purple, and a smaller chromosome. And again, we've got a blue and a purple. And these homologous pairs are the same size, the same shape, they have the same genes, but remember they have different alleles. So the example that, an easy example that isn't quite biologically accurate, but we'll just go with it, is that you can think of it an allele and genes for hair color, for example. So hair color would be the gene, but you could have different alleles, for example, for dark hair, light hair, red hair, etc. So through meiosis, these homologous pairs are going to line up together during metaphase and then be pulled apart by the end of meiosis 1. So our daughter cells at the end of this first division are going to only have a single member of that homologous pair contained within them. So our daughter cells only have one large and one small chromosome, as opposed to having a pair of large chromosomes and a pair of small chromosomes. And then meiosis 2 is just separating out those sister chromatids so that our final products of meiosis are haploid daughter cells. Okay, so actually, before I get into this stage, you'll notice that it's most clear, or it's first most clear in this diagram right here, that we have these chromosomes that have multiple colors within them. So this chromosome right here is mostly purple, but there's a little blue piece right here. And this chromosome is mostly blue, but there's a little purple piece right here. So this first mechanism that we're going to be talking about is called crossing over, and it's why you're seeing the chromosomes depicted in this way. Crossing over occurs when homologous pairs of chromosomes exchange equal segments of DNA. So we're going to have pieces of homologous pairs detach and then reattach to the other member of the homologous pair. And the result of this is we get new combinations of alleles, and these new combinations are called recombinant chromatids. So chromatids that have a combination of maternal and paternal genetic information. So I think this is easier to visualize by looking at this diagram down here. And while I'm explaining this example, I'm just going to say that this more pinkish colored chromosome on the left is the maternal one, and this more purplish colored one on the right is the paternal chromosome. And it's not entirely accurate to use the examples that I'm going to give, but just for ease of imagining things, I'm going to go with hair color and eye color as two traits that we might think about. So let's imagine that down here, close to the centromere, we have a gene for eye color. And let's say your mom says that you're going to have brown eyes. Your mom's chromosome says brown eyes. And then out here towards the tip, we have an allele that says it's a hair color gene, and it's an allele for brown hair. So your mom's chromosome says brown hair and brown eyes. And on your paternal chromosome, you have the same genes. So you're going to have an eye color down here. But let's say that your dad's chromosome says that you're going to have blue eyes. And the hair color gene that it'll also have says blonde hair. So we've got the same genes, hair color gene and eye color gene, on the maternal and paternal chromosomes, but they're different alleles. So they're saying different things for what kind of trait you're going to have. This middle picture is showing the actual crossing over event itself. So you can see the arms of the homologous chromosomes are crossing one another. That's where the name comes from. And then pieces are breaking off and attaching to the opposite chromosome, the other member of the homologous pair. And the result can be seen over here. So we can see the maternal chromosome now has some pieces that were originally derived from the paternal chromosome, and vice versa. The paternal chromosome has some pieces from the maternal chromosome. And the result of this is that now we have new combinations of alleles that didn't exist before. So we said that the mom's chromosome originally said brown eyes down here and brown hair up here. But now, because of this crossing over, this side of the chromosome is going to say brown eyes because of the gene down here that's maternal, but it's going to say blonde hair because of this piece up here that's paternal. So we've got a new combination of brown eyes and brown hair, and now brown eyes and blonde hair. And the same thing is going to happen on the paternal chromosome. Paternal chromosome originally said blue eyes, blonde hair, but now this side is going to say blue eyes and brown hair. So we're getting new combinations of alleles in these recombinant chromatids that didn't exist originally. And so the result of this is that when we go through, this is all happening during prophase 1, right when the homologous pairs are finding one another and getting ready to line up along the metaphase plate. This is when the crossing over and swapping of pieces is happening. And so the result is that by the end of meiosis, depending on what sort of chromatid the gamete gets, it could either have totally paternal information, totally maternal information, or a combination of both maternal and paternal alleles, depending on what chromatid the daughter cell gets. So that's crossing over. And the next one is called independent assortment. So this also has to do with homologous pairs and what they do during meiosis. Independent assortment refers to the fact that homologous pairs are going to line up during metaphase and separate into different gametes randomly. So they do it independently of what the other pairs of homologous chromosomes are doing. And this is illustrated over here to the right. So here, we're only showing two homologous pairs. I'm going to say blue is paternal and red is maternal. So here, we've got a homologous pair of larger chromosomes and a homologous pair that's smaller. And this one possibility shows the paternal chromosomes all lining up to the left side, and the maternal chromosomes also lining up on one side to the right. So when we pull these homologous pairs apart, one daughter cell is only going to get the blue paternal chromosomes, and the other daughter cell is only going to get the red maternal chromosomes. And then we'll follow through meiosis 2 and get gametes that only contain paternal and maternal information, respectively. But it's also totally possible for the chromosomes to line up like this. So for the paternal chromosome number 1 to be on the left, the maternal on the right, but then for the second chromosome to be the other way around, have the maternal on the left and the paternal on the right. And if you look at what happens to the daughter cells when they line up like this, you can see that they're different. Now we've got a daughter cell that's got paternal chromosome 1 and maternal chromosome 2. And this other daughter cell has maternal chromosome 1 and paternal chromosome 2. So the end result is that the gametes get a combination of maternal and paternal chromatids or chromosomes. So this seems a little trivial when we're only looking at 2 homologous pairs, but remember that humans have 23 homologous pairs. And so thinking about all of the different combinations that can result from each of those pairs lining up in either orientation with either the maternal or paternal chromosome to the left or to the right. So we can get quite a lot of variation as we increase the number of chromosomes that are independently assorting from one another. And then finally, and I think probably the easiest one to conceptualize, is outcrossing. So this just refers to having more than one individual creating offspring. So outcrossing is when gametes from different individuals combine, and this results in greater genetic variation because you get the combination of the genetic information of 2 individuals as opposed to just one. And this can be imagined with flowers that either self-pollinate or cross-pollinate. So a self-pollinating flower is going to make both the eggs and the sperm. And so you're only getting the information from that one flower in its offspring. Whereas with cross-pollination, one flower is going to make the sperm and another separate flower is going to make the eggs. So their offspring is going to be a combination of both of their genetic information. Okay, I know that was kind of a lot to absorb, and I think that the importance of all of this will become more apparent later, but hopefully independent assortment, outcrossing, and crossing over, at least the foundations are there after this video lecture. I'll see you all soon.